Okay, last lecture, I basically got various odds and ends, and I'm not even sure I should have brought you here, but I did. We did. So here's what we're going to do. You want to say something before I start? Okay, first of all, I've got a few minor things and leftovers I want to talk about briefly. And then I want to talk about our agenda, now that you've all worked out the circuit from here on through. Some of you will have already started doing this. I don't know where you are in this now, but you're probably, at least some of you are not too far along. Probably most of you aren't too far along. And then I want to, I have a brief, I want to indulge in a little bit of true psychology for a minute as opposed to physiological, as opposed to behavioral neuroscience, to ask you some questions. Okay, first of all, the minor points and leftovers. First one's very easy. Well, it's very easy. If I can. Oops! Happy Halloween. Right. That was not it. That was actually why we had to get here. You had to be able to see this picture. Okay. That's. Come on. There's what I want. So somebody said to me last time or the time before, Swimmies neurons don't have after hyperpolarizations, after the spike. And I was pretty sure that was wrong. But I figured I'd better check, and it was wrong. So the reason you don't normally, so here's the after hyperpolarization, here's a spike that I caused by injecting some current into the cell. Then here is the after hyperpolarization. It's not huge, but it's there. If the cell were, for one reason, sitting up here at a higher baseline potential, then it would look bigger. The reason you don't notice it in most of the traces that you have is there's all kinds of stuff going on all the time anyway, and you don't know what's after hyperpolarization and what's an IPSP and what's the recovery from a previous EPSP, or because there's continual activity on the baseline when swimming is normally going. But in the cell, like cell 4 in that circuit we used the first week to demonstrate various things where you are actually, the cells are perfectly quiet when you're not doing something to them, so you really know where the resonant potential is. You can see there is, in fact, an after hyperpolarization. And that's due, as you all know, to potassium coming into the cell. Okay, the other thing is actually from the first lecture, which I didn't... something I think I didn't quite get to, and Dr. Grisham might have done this in my absence. I'm not... You haven't? Okay, right. I just want to say a few words about some things here that will help you with your lab reports and so on. So during the first week, you had to demonstrate depression and facilitation. And so here, I may or may not have already showed this slide. Here is the baseline EPSP in the case of the depression experiment. And then if you give a second whack to the presynaptic input, to the input that was causing this EPSP here, somewhat after the first one is over, so okay, the first one goes like so, up, down, and it's pretty much over there, and then we hit it again, and it's obviously smaller the second time. And that's an example of depression. We urge you in your lab reports not to do it like this. We could have given the second stimulus soon after the first one, in which case it looks like that. There's several problems with doing it this way. One problem, which isn't really very great in this instance, is that in this case, there's if at this point in time here we had simply had this EPSP. So this guy here is obviously quite shrunken. This is even smaller than it was here, and I'll come back to why that's so in just a moment. But if that had not been so if when we hit the thing here, the EPSP here had simply been the sum of that EPSP and this EPSP, and suppose it had been the same size as this one. In other words, we have the big one and then one a little bit smaller. But the second one had been riding on the first one. If you just look at it, it's hard to tell whether it's bigger or smaller when one's riding on the other. And you can figure it out. You can make some measurements and figure it out. But it's not a good way, as Dr. Grisham says, don't make your reader think if you want a good grade. It would be a bad way to, it would be bad to put it right there because it's kind of hard to see what's what. Whereas if you put it here after the first one's over, it's perfectly conspicuous that the second one's smaller than the first one and you're home free. but there's a deeper reason why it's a bad idea to put it here. This one is smaller than this one. That could be for the most. If I asked you why is that so, what you probably would say is, well, it's sooner after, so it got tired out and it had more time to it partially recovered here. If I'd waited a long time, it would have been fully recovered. And here, since it's very soon after the first one, it's really tired out. There would have been an element of truth to that, but it would not have been the whole story. And the reason it wouldn't have been the whole story is that or I've told you tried to explain to you several times, and I hope you know now, that when an EPSP rides on an IPSP, it is shrunken because of the shunting action, the shorting out action of the IPSP. Well, that shorting out action is simply due to the fact that various ion channels are open, due to the inhibitory transmitter. During an EPSP, ion channels are open too. Different ones, but they're open, and that allows things to short out through the membrane. So in actual fact, even if there's no depression at all, even if this guy was the original size, if we put it here right after, while the first one was still going on, there's a pretty good chance that that the first EPSP would partially short out or shunt the second EPSP. So it kind of like an inhibitory action, a little bit. And so the shunting thing is not just something that IPSPs do. Any synaptic potential does it. Anything that opens the ion channels will cause that to happen. That, I think, is all we really need for that slide. Okay, now. Okay, first of all, Dr. Grisham often asked me to go over this, and it rose in one of the sessions during the week, but not the others. So I just want to expose you all to it. Just clarify things. So I think you all know by now what endogenous means. Literally, I looked up some definitions, and the one I liked best was arising from within. I got that off of Wikipedia or something like that on the web. We've talked about two different kinds of endogenously active cells. One kind is what we call tonically active. One kind is what we call bursting. These are fairly common terms used in the literature by other people, pretty much the way we've used them, but they're not always used exactly the same way. And just so the important thing for us right now is that we all use them the same way, because you're going to be writing your lab reports, and we don't want you to get marked down because you use the words in a way different from the way your graders are going to be using them. So in here, tonically active means regularly firing. Strictly speaking, tonically active just means always active. But we want to make a distinction between activity which is regular or pretty regular and activity which is in this brr, brr, brr pattern. So we've distinguished between tonically active cells and bursting cells. And the term bursting is not universally understood, but is often understood. And sometimes cells like that are, for reasons that I won't bother explaining, sometimes called beating cells. I know where that comes from, but it's kind of bizarre. I mean, it's arcane, and I won't bother explaining it. But anyway, so bursting cells are ones that go brr, brr, brr. Tonically active cells are ones that go brr. And either of those things can happen either because they get input from somebody else or because they are endogenous. So if you know a cell is tonically active or bursting, it doesn't necessarily mean it's endogenously active. It might be doing those things because of input from someplace else. So the endogenous aspect of it means that the cell itself does it on its own hook. Okay. Now let's progress to what I actually wanted to be what I meant to be talking about this morning. Once you know the circuit, your job is to try to figure out how it works. And you can, as we've told you, I think quite a few times before, you can assume that it's one of the three mechanisms we discussed in the lab manual. That's our gift to you. There are other variations that people have thought of, but these are the most important ones, and they're the only ones you need to worry about. In doing that, as I told most of you during lab, I didn't say exactly the same thing to all the different sections. Your first a good strategy to start with, since what you really want to know is what the mechanism of rhythm generation. That's the most important question. There's some other questions that you want to figure out too, but that's the most important one. You want to see if maybe you can break the circuit up into some cells that are actually involved in generating the rhythm and other cells that are not involved in generating the rhythm and are simply following the ones that are generating the rhythm. And that's a distinction we obviously made in all the groups because we made a big deal out of trying to figure out which were the generators and which were the followers. And you're going to have to do that too, and you can either do it, I guess you can do it the way we did it in class, or since you got all the cells, there are a lot of other ways you can do it. Then once you know which is which, if these guys here maybe are the generators, you'd want to study them to figure out how they're generating the rhythm. We'll come back to that. So here's a little more guidance on that. Okay, first of all, as I said in class, this goal can be oversimplistic. Some circuits, all the cells are involved in generating the rhythms. Now, in actual fact, usually motor neurons, that's 1 and 2 here, are not involved in generating rhythms in real animals, but of course swimming is not a real animal, despite the fact that we've all become very fond of it, no doubt, and may think of it as a real animal. It's not. And so what's true for a real animal may not be true in swimming. So you can't even be guaranteed that the motor neurons are not part of the rhythm generator. That's for you to find out. So the goal can be oversimplistic, and all the cells can be involved. And I may or may not give you an example in a minute of a real case where all the cells are involved. Okay. So as I say, it's worth trying to make the separation. You can do it the way we did it in class this week, or another strategy here where you know all the cells would be to silence some cells and see what the others do. So if you silence a bunch of cells and the other cells go on exactly, you keep doing the thing perfectly, probably those cells you silenced were not part of the generator of the rhythm. On the other hand, if you stop some cells and that mucks up the rhythm, those cells are very likely to be part of the generator of the rhythm. So that's the kind of technique that you can employ. Just out of curiosity, how many of you already know which cells are generating the rhythm in your swimmies? Raise your hands high. I'm just curious. How many do not yet know? Yeah, okay. Okay, the heart's a case of one where all the cells do it. I don't think I want to bother talking about that. Okay, once you figure out which cells are generating the rhythm, as I said, you'll have one of those 3 mechanisms because we were telling you that. You will try to figure out which of the 3 it is. Now, let me make some points about that. First of all, we've just given you 3 mechanisms. And so one approach is to try to eliminate 2 of them and say, okay, obviously it's the 3rd. That's not quite good enough. If you just do that in your lab reports, you won't get very good grades. You're going to have further evidence that the one that you home in on, it really is that one. I'll come back to that point in a few minutes. But another point, and this is the point I really want to make right now, is in the lab manual, and for that matter in the study questions, we assumed, we do certain circuits as illustrative of certain mechanisms. What is general and what you can count on is that one of the basic ideas or kinds of mechanisms are what are generating the rhythm in swimming. But you cannot be guaranteed that the circuit will look exactly the same way. So, for example, what does that look like of the 3? If you just saw that picture and you said, just Clang association, which of the 3 mechanisms would you guess, if just looking at the circuit would be your hunch, your best bet? Yeah, it looks kind of like it looks like the picture we drew for mutually depressing inhibition. Correct. Suppose instead you saw that. Could that be mutually depressing inhibition too? How many say it could? How many say it couldn't? Okay, it could. Won't this time force you to answer. What's essential about this idea of mutually depressing inhibition is the 1 and 2, the 2 cells on the 2 sides have to be endogenously tonic. Or, actually, they don't even have to be endogenously tonic. They have to be tonically active. Something's got to make, maybe they've got to be endogenously tonically active, or they may be getting input from somewhere else that makes them tonic. It doesn't make any difference, but they have to be tonically active. And they have to inhibit each other. Either, it doesn't really matter whether they inhibit each other via an interneuron or directly. And something about that those inhibitory links has to be able to tire out and then to recover with rest. And whether in this circuit, there's only one thing that would tire out. It would be that synapse there. But in this circuit, it could be that synapse and it could be that synapse. And you would get rhythmic, you would get bursting out of this, alternate bursting either way. It doesn't matter. So you're looking for so you need to understand the mechanism and then trying to see if that mechanism seems to be operating in the pattern generator, in the generator of your SWIMI. But you cannot slavishly look for the same picture. So for example, this picture here, where else do you see that? And so where do you see that in SWIMI? Where's one place you see that in SWIMI? In fact, maybe it's the only place you see it in SWIMI. Remember, you've all just worked out the circuit. You have all done this already, right? Everybody's done. We all know this, right, Bill? Yeah, we all know the circuit. Okay. I don't want to give anything away here. I think that's it. All right. Where do we see that pattern? Right down there, between the two motor neurons. If you looked at that and said, "Oh, that's obviously a mutually depressing inhibition oscillator," you might be right or you might be wrong, but you bloody well better check it. Okay, so that's one example of what I mean when I say you can't necessarily just count on things based upon the way they look. You've got to go a little deeper and ask how do they actually work and what's the concept at issue, not what's the particular anatomical picture at issue. That same thing applies in other cases in the lab manual. So for example, this is the, I think on the left, I think, is the one example we use in the lab manual to illustrate mutually to illustrate the ring oscillator. Here's the ring, and it's driving a left and a right moving muscle for the tail fan as it goes back and forth. If we could instead, the ring here is the same, but we have that driving, and this part of it's the same, but now we have a tonically active cell here, which gets inhibited, and that's driving B. This circuit would work, if you just looked at what A and B were doing, this circuit would do pretty much exactly the same thing this circuit would do in terms of the A and B, which for those of your motor neurons, it would be producing perfectly fine swimming. But the pictures, the anatomy of the two circuits is obviously different, but the concept is still the same. It's just employed in a somewhat different way in the two circuits. So you've got to watch out for that when you're trying to work all this out. Now, oh hey, we're really getting along here. I'm sorry, I got to remember what I want to do here. Oh, right, I see. Okay, all right. Okay, now, this is not swimming, but suppose we had a circuit like this. Suppose you found something like this in swimming. Suppose you found something like this in swimming. You've got two neurons coupled by that are tonically active. They're coupled as shown. This synapse here on each side shows short-term depression. But the reason that B and C are tonically active is not because they're endogenously tonic, but because neuron A, which is tonic, is driving both of them. Okay, question one. Would B and C show alternating bursts of activity? Would they be basically do the same kind of thing that SWIMI does? How many say it would? How many? Oops, sorry. Wait, sorry. How many say it would do it? Same thing. How many say it would not? Okay, you're not all voting, which is a good opportunity for me to do something I want to do. I want to get some votes here on something. I'm very curious. This has got nothing to do with SWIMI, and it's got nothing really to do with you. It's for some other issues that I am concerned about. It's really hard to get you guys to vote. You're all getting better at it. but you're far from perfect. I have to badger you almost every time. Occasionally I don't. When I get some of your groups are a little better than others, but when you're together in this big room or when you're in the smaller group, it's a little hard sometimes. And I would like to find out, and you may not be willing to raise your hands on this either, but this time I'm going to try to insist. I want to try to find out why. And you can vote for more than one of these things, but let me put them down in big. So one is that you're being mentally lazy. You just don't want to sit here and think, so you don't want to bother. You know, it's easier just to sit and listen and not think. Huh? That's number one. Yeah, well, I still want to see what people say. Another is that you are afraid. And there are various things you may be afraid of. See, I'm going to be a real psychologist when I want to. One is that you're afraid because you're going to have an ego blow if you're wrong. So it's your ego that you're afraid of. One is that you're afraid of each other. And the other and final one is you're afraid of me. Okay, now then, you can vote for more than one. But I want to see lots of hands if it pertains to you. How many, for how many is number one going on? The mentally lazy. Raise your hands real high. Okay, I thought that might be the case. Is that Bill? Bill, shame on you. Okay, now, ego. How many? Not very many. At least not very many admitting to it. Okay, each other. Me. So how many of you? Okay. So you've all admitted to this. So I just want to get the broad. Never mind which of these things. For how many of you is it some sort of fear of embarrassment or that is keeping you from raising your hands? Raise your hands very high if that's the case for you. For how many is that not the case? Okay. So most of us mentally lazy is a big deal. Okay, that's very interesting. I'll tell you why it's interesting and why I have a variety of reasons for wanting to know. But one of the things that we are thinking of doing in this department, some of us at least, is how many of you have had classes where you use clickers? Well, we're thinking about doing it some, and what you just told me, I'm sorry, what you just told me suggests that clickers are a very good idea. For those who don't, what? Yeah, the problem is you probably don't all own the same kind. Yeah. Those of you who, I heard big groans. How many? So for those of you who hate clickers, sorry, is there anybody who likes clickers? You like them. That's right. Oh, look what you did to her. She using see now that was each other. You made her she put her hand down because of that one. She was afraid of the social pressure. Thank you. For those of you who hate them, I'd just like to hear why. Oh, you mean technical reasons? Okay. Are there other reasons? How much do they cost? $50. Okay, I think you can get them a lot cheaper now. But anyway, yeah. Okay. Are there? Huh? You forget it, you lose credit. Right? Yeah. Okay. Yeah. Okay. Huh? What? Yeah, that's right. Well, I think probably the big problem is that it's very. That's the point. That's why I'm thinking that's a great idea. Okay. Okay, thank you. That was very helpful. That actually was very useful to me. Okay, let's get back to business here. A lot of you, I mean, so far as you all, some of you didn't vote, and I didn't push it too hard, but a lot of you thought that B and C would not do this, would not burst, alternate, not do the perfectly, but they would. They absolutely would. You should think that through because you should, seeing that it will work, will help ensure that you understand this mutually depressing inhibition mechanism. It doesn't matter what makes B and C go, as long as they have a tendency always to go, which they do here if A is driving them both all the time. What will happen is whichever one's going will be shut off by the other one until that inhibition tires out, and then the other one can go back and forth. So that's very important to understand. Okay, so would A show any alternating bursts of activity? No. If we hyperpolarized A, what would happen? Would it stop? Yeah, sure. Which cells here are endogenously active? A, only A, right. Now, in our way of finding neurons that were involved in swimming, given this, what we told you to do, you wouldn't have found A. I mean, you wouldn't have identified it as being a swimming thing, because we said the only cells that are probably, all the cells that are involved in producing swimming are probably active in some phase relationship to the alternating bursting, and that's not true of A. It just goes all the time. Would B be part of the generator of the rhythm? Yes. How many say yes? How many say no? Okay, the answer is yes, it would. We're not asking what are the sources of endogenous, and this is very important for your getting your lab reports so that you get good grades. When we ask what is the generator of the rhythm, we're not asking just what is the source of endogenousness. We're asking what is the source also of the rhythmicness. And whereas it's true that in this case A is the source of the endogenousness, the rest of the circuit is what's responsible for the fact that it goes absolutely, all the cells are involved, I think it would be fair to say, as being part of the generator of the rhythm. Well, and then I said with A, which tries to be part of the generator of the rhythm, and I've just now answered that for you. The answer is certainly, well, is A part of the generator of the rhythm? It's not part of the, maybe you might say it's not part of the generator of the rhythm, but it's part of the generator of the activity. I think if you had to address that question, you'd want to say exactly what you meant and why you meant it, and not worry too much about definitions, because there might be some definitional issues there. If you ask me, I'd say yes. I think say the package as a whole is what's generating the rhythm. That's the circuit that's generating the rhythm. And muck up any part of it, and you don't get the rhythm anymore. All right, well, I actually didn't think I should lecture because I thought I'd already told you everything you needed to know, but from this discussion I realized it was a good thing I did this because it'll help you with some of your reports. Then the last point I want to make, oh no, not quite the last point. How are we doing? Okay, 5 more minutes. I got to get out of here because Bill has some other things he wants to do. So as I said a few minutes ago, you can't, for example, just decide what the mechanism is by excluding the other two. Another way of saying that is once you think you know what the mechanism is, you've got to try to marshal all the evidence you can that you're correct. So, for example, if this was a mutually depressing, and if you decided that these motor neurons were in fact coupled by mutually depressing inhibition, these connections really, that 1 and 2 were endogenously active and so on and so forth. Or were driven by C and D, maybe. I don't know. Yeah, they could be driven by C and D, which are also, well, I mean, oops. Anyway, let's start that again. I was giving things away. If 1 and 2 were endogenously active and you decided this was probably a mutually depressing inhibition oscillator, what are the things you'd want to have to show in order to get a good grade on your lab report? One thing is you'd have to show that 1 and 2 left to them when 2 was gotten rid of, 1 went all the time, and conversely. You'd also have to show, and this is what people would tend to leave out, you'd have to show that this pathway here really did tire out. You'd have to show the depressing part in some way. And you'd probably also need to show which of these 2 synapses was the one that tired out, that one or that one. Having done all that, you'd be in pretty good shape, but we'd expect you to do it all. So, and in particular, doing it by exclusion, saying it's not the other 2 mechanisms, therefore it's this one, is definitely not okay. It's a step in the right direction. It can give you the answer, but it won't get you a good grade. Now, last point, which Dr. Grisham has already made, but I want to make it once more. Suppose that you get all through, and you think you've got it all worked out, and so on and so forth, or suppose this happened in real life. You decided maybe you found all the most important neurons and connections and figured out how the circuit seemed to work. How would you know for sure that there were not still more connections or neurons involved and that you had not in fact found the real way that it works? Maybe your circuit is just an oversimplified version that is just a little part of a much more complicated true reality. How would you know? And the answer is you wouldn't. Okay? But it would not be a good use of your time to keep chasing your tail unless there was some reason to think there was something wrong. And you would therefore use, and this is the principle Dr. Grisham told you each about this last week, Occam's razor, the principle of parsimony, which is the philosophical principle that says if there is a simpler and also a more complicated way of explaining the same thing, then the simpler one should be accepted as the true one. But with the recognition, of course, that all ideas about the world are provisional, and something could come along that might force you to change your mind. Okay? Fair? Did I do your ancestor due justice? Bill often claims that Occam was his ancestor. I don't know if he's still saying that. But I think that's everything. Yeah, we don't need this last thing. Oh, uh-oh. Yeah? It shows that it is a part, an integral, an essential functioning part of the circuit that generates the rhythm. We have a bad habit, and I pointed this out in some of the sections, but not all of them. We have a bad habit of talking about neurons as generators. That can be correct sometimes, but in most cases, it's a circuit that's the generator. Individual components of the circuit may, for example, be endogenous bursters, but that may not be the whole story about the rhythm generation. So, for example, we talked about the idea that if you have a circuit that's driven by endogenous bursters, and if in a bilaterally symmetrical animal, if you've got a burster on one side and a burster on the other side, that if they are totally independent of one another, you're going to have problems because gradually the two sides will get out of appropriate sync with one another. So it would be fair to say that in a certain sense that it is, so therefore the two sides have to somehow mutually inhibit each other. And it would be fair to say that it's the whole circuit, the two endogenous bursters and the mutual inhibitory link as a whole that are the generator. Now, if that were the way it was, and you tried, well, that's right. If that's the way it was and you tried to do the resetting experiment, it would probably work. But the fact that the resetting experiment is like virtually all experiments in science, is great when you get positive results, and it doesn't mean much when you get negative results. Because if you are able to reset the rhythm, the timing, in other words, after you hyperpolarize for a little while to stop the firing, then you let it go, and if you find the timing of the subsequent activity is different than it would otherwise have been, that is proof positive, I think. One hates to say something like that, but that's proof positive that this neuron was part of the generator. But if that doesn't happen, if you can't reset the rhythm, it does not prove that it was not part of the generator, because it just may be that manipulation was not just done in such a way, and in fact that it worked. And in fact, in some of the sections, we picked a particular time, at least initially, we picked a particular time for doing our hyperpolarization, so we didn't get very much effect. And I said, let's try a different time when we'll get a bigger effect. Well, you could imagine you might be able to pick a time when it had no apparent effect at all. So in science, in this instance, in this case, negative results are very, you have to really be careful about drawing inferences from them. And that's true in all of science. You basically can virtually never get a paper published as a scientist if you fail to find something. You have to find something positive, because otherwise, you never know. If you just done it a slightly different way, you might have found something, and so on and so forth. Okay? Can I go? No. Oh. Yes?
All right, so you've shown us all sorts of wild things in this model. You've shown us tonically active cells. You've shown us bursters. You've shown us cells that depress and cells that facilitate. And my question for you is this all something that you came up with in some hallucinogenic haze, or do these things really exist in real nervous systems?
All those things happen in real nervous systems, for sure. Lots. For example, depression and facilitation. Now, I don't know this for a fact, but I'd say that probably this is probably an overstatement. But I'm not sure. Let me put it this way: I'm not sure I'm wrong about the following statement. It's almost impossible to find a synapse that doesn't show at least a little bit of facilitation, or a little bit of depression, or a little bit of both. synapse that doesn't show that. Now, that may be an overstatement, but they're very common phenomena. These mechanisms, the three hypothetical mechanisms we're talking about here, are all used by certain animals to generate their locomotor rhythms. Leeches do the ring thing and also some other stuff, I think. Lobster heart, which is the one that's been studied the most, or the lobster stomach, which is the thing that's been studied the most, and is the one of the cases in the world that you can kind of do the way we do swimming, which is find all the neurons and figure out how they work, uses both endogenous. It actually has two different rhythms for one part of the stomach, another part for the other part of the stomach. And one of them is based on an endogenous burster, and the other one's based upon mutually depressing inhibition, or something kind of like it. Works a little bit different way than ours, but okay.
Okay, and then lastly, I did tell you that Dr. Krasne did recently win an award.
Oh yeah, but I haven't seen it.
I actually now have it with me, so here it is. The Editor's Choice Award for Outstanding Neuroscience Laboratory Article.
That's the good news. What he's done. Oh, I thought you did too, right?
Yeah, but I got my own certificate.
Oh, I see. You got it in front.